Yeah, so we typically do not have commercial speaking sessions here at the Go Ahead Exchange. However, we thought that the audience would find interest in these next innovative sources. In this session, we'll highlight 3 unique omega-3 sources: herring roll, Calanus phenmarticus, a genetically modified canola omega-3, as well as a presentation on a new unique technology, lysophosphatidylcholine. Okay, I want to bring up our first speaker. Høgne Halleraker is a Norwegian entrepreneur from the life science industries. He has founded several companies and is currently the CSO of Arctic Bioscience in Norway, a company that extracts omega-3 phospholipid-rich oil found in Norwegian herring caviar. This oil has been sold commercially in the nutraceutical space since 2012 and is now being developed as a pharmaceutical drug to treat psoriasis. Halleraker is currently also the chairman of the board at NCE Blue Legacy, a Norwegian organization that aims to improve the use and profitability of marine resources and establish new projects and businesses within fisheries and adjacent commercial opportunities. With that, we welcome Høgne Halleraker.
Thank you, Mike, the great biker and fly fisherman of Colorado. Okay, I'm glad to be here. Hi, everyone. I'd like to share with you some of the stuff that we are doing in Norway at Arctic Bioscience. I'll go through a little bit about who we are, about the nutraceutical stuff we do, a little bit about the science, and then at the end, the results of our pharmaceutical study. So I'm trying not to waste too much time. This is a picture of Norway, by the way. It's a beautiful country. And to start with, Arctic Bioscience is basically involved in 2 different areas. One is the nutraceutical with our brand Omega. The other one is the pharmaceutical, where we don't have a brand name yet. It's called HRO 350. And as a teaser, you can see down on the right-hand corner, some picture of the results that we found using herring roll oil to treat psoriasis. This is based on a clinical study that we did in the University Hospital of Bergen back in 2019. A picture of our headquarters. I like to show that because it's a nice place, and the herring that we source is actually right outside the fjord, just about a couple hours drive. And we'd like to show our customers our headquarters and also the fishing grounds where this happens. So we typically harvest our herring 2 times a year. So in terms of the fishing grounds are very close to us. The herring is a very sustainable species. They come in typically in September and January, February to spawn, and the herring is used for its fillets. And we collect the roe and freeze it and extract our lipids from this roe, where we make our products, the lipid products, the protein products, and also our pharmaceutical starting material. My last slide will also show that we are in the process of setting up our own facility to extract these lipids, which will be very exciting, I think. It's one of the first and only pharmaceutical factories in Norway from a Norwegian product that is marine-based. So what is our product? Our product is extracted from a fish egg. The fish egg is a powerful, let's say, ingredient because it contains the recipe to create new life. So this picture here is actually from a zebrafish. And if you can look at the development stages from fertilization to the hatching of this fish takes only 48 hours, which tells you something about the potency of the fish egg. It has everything needed to generate new life, all the lipids, all the proteins, all the enzymes in the right composition. So this is the basis of our business plan, if you want. The other part is the lipids we extract. These are membrane lipids. And membrane lipids are different from, let's say, storage lipids. Triglycerides, for example, are typically storage lipids. And we operate in the phospholipid space. where we have all the different phospholipid species, the phosphatidylcholine, the ethanolamine, and the inositol serine in a certain composition. Additionally, these phospholipids also come in their lysoforms, which is also present in the product. This whole thing started with me back in the day. When I was a student at the University of Bergen, I worked with aquaculture and halibut. The halibut have issues when they want to during metamorphosis from their original fish-like swimming style to becoming a flatfish. So in this picture, this is an ordinary halibut, very healthy. These other ones have issues. Either it lacks an eye, it's white, it's deformed, scoliosis or lordiosis, and we found out that using membrane lipids in the early life stages created this kind of halibut and not the other ones. So there is some kind of, let's say, we thought there was some kind of magic in the oil, and we now know that these are phospholipid esters rich in DHA and also some other stuff I can't share at this point. A little bit about the science. Just briefly, phospholipids is what we do. Phospholipid esters is what we extract. These phospholipid esters are rich in DHA, they're rich in EPA, and this is a complex mixture of what we like to say biologically important phospholipids. And I can also say that the phospholipids, as you may know, they are pretty solid in their concentrated state, so we need to add in a carrier oil to make the product liquid. Just a few slides from what we like to say is our goodie bag. This is SPMs. For those of you who know SPMs, these are metabolites of DHA and EPA and have functions as an anti-inflammatory agent. What you can see from this slide is that all these different SPMs are grouped. These are maricins, resolvins in the E series, in the D series, and also a precursor for the maricins. So these, we think, are a key to understanding the anti-inflammatory properties of omega-3s. You can also note that we think that our SPMs are also phospholipid bound. It means that they are more stable than, let's say, other sources. Another thing I like to share with you is we did some studies back in 2016 in Singapore where there was a paper that created some attention. They found a transport protein called MFSD2A, and this protein is the one that picks up the lysophosphatidylcholine and actively, let's say, drags it into the brain. So we made this product for this study, and this is for the eye. So the blood-eye barrier is difficult to penetrate, but the lysophosphatidylcholine DHA can do it. A little bit about the studies we've done on our membrane lipids. We've done studies with pigs on brain, reducing peripheral inflammation after immune activation in a piglet model. These are all studies that are published. We also did a bioavailability study where we looked at phospholipid-bound DHA and EPA compared to other sources and found also that they penetrate membranes very quickly compared to the triglycerides that we looked at the time. Then we also did a few clinical studies, and this last one here is actually the basis of our pharmaceutical, let's say, efforts that we are into right now. Before we go on to the pharmaceuticals, as you know, in the omega-3 industry, there are a number of different indications you can target with different omega-3 products. We think that our membrane lipids are best suited to address, the ones I've listed here, the healthy aging, cognitive support, sports nutrition, and as you know, these are all in some way related through inflammation or lack of omega-3 fatty acids. So if you list these different ones, let's say the properties of what we have stability, of course, the phospholipid, the bioavailability, the source of choline, and the source of vitamin D is what we like to focus on. Then, our pharmaceutical development. This is, of course, a stretch. This is a new indication. Omega-3 fatty acids, as you know, have been used to treat severe triglycerides or high triglycerides. And this indication is something new. And it was based on this result that we got from the Bergen study. This is actually patients. We have 30 patients on treatment, 30 patients on placebo. Duration is 26 weeks, and it was elongated to 52 weeks. And we got some very interesting results. And we saw that with this small group, we got a significant result on the treatment of psoriasis, mild to moderate psoriasis, with this HRO 350. If you look at the graph, I'll try to explain this. The yellow is the placebo until week 26. Then all the patients were tired of the placebo. They wanted our product instead. So we switched them to our product. They instantly went down here and met with the other group who's been going on the product since the beginning. This is measured as PASI. PASI score is a psoriasis area severity index. Tells you something about how much psoriasis you have and also how much you get rid of when you do the measurements, let's say, in different timelines in the study. We also wanted to know if the effect of the product went away after a while. It didn't. It was the other way. It improved. So over time, the effect of the treatment improved. So after week 65, almost 40% of the patients had clear or almost clear of psoriasis, which is a very strong result in this kind of study. Also significant numbers, which was, to us, of course, very pleasing to see. This PSGA score is basically a physician's assessment of how the product performs in a patient. If you look at some other secondary outcomes of this trial, we looked at the DLQI index, which is the dermatological quality of life, which also improves, meaning you ask the patient, does this mean anything to you? And yes, it does, very much so. Improve their life quality very much. One of the greatest benefits of using natural products is safety profile. In all pharmaceutical development, you have safety and efficacy as two major issues. For us, using a natural product extracted from a natural resource, we think that we have an advantage to become the first-in-class oral treatment for mild to moderate psoriasis, meaning that any kind of patient comes in to the doctor's office, I got some psoriasis, what is the first thing I should try? Well, HRO 350, because it's safe and efficacious, right? So, let's say, round off this pharmaceutical presentation, this has opened up to us a whole range of different possibilities and opportunities. And of course, these autoimmune diseases are related. And if you look at the slide here, you probably recognize a number of them. And we have anecdotal evidence, if you want, on quite a few of these, because we think that the mechanism of inflammation is pretty similar in many of these diseases. And we hope that we will be able to target also other indications. We do have a program going on now with extreme premature infants, where the key, let's say, goal is to give nutritional support to extreme preterm infants born before week 28 to help them develop their brain, let's say, in a better way than today, because they don't have anything to help them with. So that's also something we look into. Let me see. Oops, that was my last slide. And this is our manufacturing facility that we are now planning. This is in Norway. It's adjacent to this building in the back. And by doing this, we were able to develop the product commercially, let's say, or in commercial scale, not just lab, not just pilot. And it enables us to, let's say, take control of the whole value chain on the both nutraceutical and the pharmaceutical side of things. Yes, so this has actually been a goal of mine for many, many years, to have a pharmaceutical factory in my hometown, so to speak, based on a Norwegian natural product source from a sustainable fishery. Actually, it's a side stream, and back in the day, it was used for animal feed. So, yeah, and that's pretty much what I wanted to share with you. So, thank you.
Thank you, Hayden. I forgot to mention, we're going to have Q&A after the four speakers speak, so they'll come back up, and if you have any questions, you can ask at that time. Yeah. But hey, thank you, Hogne.
Sure.
Our next speaker is Elise Marie Pedersen, PhD, is the head of science, human health at Zuka, and is associate professor at the Arctic University of Norway, UiT. In the company, Dr. Pedersen leads and coordinates research and development projects unraveling the unique biomedical properties of products derived from Calanus finmarchicus zooplankton. Zuka provides novel findings in the field of omega-3 biochemistry and has shown that nature packs omega-3s in the most amazing and exquisite ways, impacting human health. I introduce Elise Marie Pedersen.
Hi, everyone. I've been talking so much these last couple of days, so I don't have my voice anymore. But thank you so much for the invitation, and Ellen gave me this easy task of condensing exactly 20 years of R&D into 10 minutes. So I'll try to limit my info. So the story is about Zuka, our company. We've developed an entirely new value chain based on this tiny crustacean floating around in the Norwegian sea currents. So this is our backyard right now. This is actually just a short walk from my house, and I heard that Hogne also showed you some beautiful Norwegian pictures, so it's kind of the same scenery. And this group of enthusiastic people, that's Zuka, and we live, as my kids complain, very close to the North Pole. So I haven't sent any pictures home, so they know where I am. And we live above the Arctic Circle, our headquarters in the city of Tromsø, and our production facilities and subsidiary in the city of Sortland. So this is a very common sight to see in the spring and summer in the northern Norway shores. Notice we still have snow, though. 20 years ago, this professor, Kurt Tande, he had spent his entire academic career studying this tiny red zooplankton called Calanus finmarchicus. And he knew that there were nothing from the literature indicating any commercial utilization of this resource. But he had this intuition that there had to be a valuable potential here, because the species had an enormous biomass and a really high lipid and protein content. So there had to be some potential. So, Calanus finmarchicus. It's an amazing creature. It's nearly at the bottom of the food web. It links the phytoplankton with the other trophic levels. It's extremely small. It's as you see, 3 to 5 millimeters, and so large in numbers. And each spring and summer, it swarms in the upper water layers, and where it blooms, grazes on the blooming phytoplankton underneath the midnight sun. It is short-lived, it renews rapidly, it has only 1 year life cycle. And when the autumn comes, it sinks thousands of meters down to the bottom of the ocean, where it goes to a hibernating state. And to be able to do this, it actually has this unique way of packing its energy. It's converting its food into these very compact lipids. And it's one of the most numerous animal species on the planet. In the Norwegian Sea only, we have this amazing figure here. This is an estimate for only the Norwegian Sea, this yearly renewing biomass. And that's actually almost double the amount of the total of all the fishery and aquaculture in the world. And this small speck here, that's our harvesting quota. So sustainable fits very well here. But it's not an easy task to handling something so small, so different in composition than anything else seen in the industry. So we started from scratch, developing harvesting technology, our production technology, analyzing new techniques of analyzing, but we managed it. So now we actually, in the spring and the summer, we very gently harvest the upper water layers. We freeze the raw material immediately, and then bring it on shore on our doorstep here in the city of Sortland. This is our brand new production facility. Some of us actually take our morning swims here all year through. And on the inside of our new factory, we do this very mild extraction of the lipids. No solvents, no additives, no refining. And the oil has a very robust natural stability. And all the biomass is used. During our process, nothing goes to waste, so the only water is removed. We have product streams for both human and animals, but so far we have most focus on the oil. So, 1.0% astaxanthin. That can sound quite small, but in reality, that's a really high content of astaxanthin. And it makes the oil almost impossible to work with in the lab. We struggled so much just to analyze. But when we finally cracked the code of analyzing this oil, it showed a very broad lipid profile. It's actually, it's a cocktail of different monounsaturated and polyunsaturated long carbon chains. And when you look at these figures, only half of the oil is actually fatty acids, and less than 25% is actually omega-3 fatty acids. And we have the most of this odd, short fellow ceridonic acid. And we still have really exciting results when we see, when we test the omega-3 index and study bioavailability. But we also know from the literature that the MUFAs can affect the biosynthesis of omega-3 fatty acids. So we assume we have some kind of booster effect of this fan of different fatty acids. And the second part of this oil, I think this is the most exciting part, is the MUFA equivalents just in an alcohol form. And these are actually oxidized back to fatty acids and utilized by the body. So, some nerdy stuff. I think this is kind of the magical part of this oil. because the zooplankton packs its fatty acid in a 1-to-1 ratio with fatty alcohols, or polycosanols as it's also called. And that's the definition of a wax. So it's the same lipid class that you actually find outside leaves and fruits and nuts and so on. And it's amazing. So this tiny animal has actually been able to condense its energy and store the fatty acids as compact as nature actually allows. And this is what gives it some special features. And in addition, it actually toggles on both the chain lengths and the saturation of the different acids and alcohols, giving it some density features also. So the animal is actually able to move along the water column just by shifting the crystallization of the different lipid components. So this is the reason why the animal actually can sink to the bottom of the ocean during the winter hibernation and stay there without using any excess energy, and then resurface again in the spring when the sun is back. So, wax esters, they are on the opposite scale of the phospholipids. They are extremely hydrophobic. So the digestive enzymes need to do a lot more work, to work a lot longer and able to cleave the different fatty components. And this is what we see as kind of the key feature of this oil, is that it lets the oil travel much longer in the intestinal system, where it actually reaches the later parts of the small intestine. And there it comes in contact with these specialized free fatty acid receptors. And these free fatty acid receptors are a part of regulating this glucose lipid metabolism. And why would there be such fatty acid receptors at the very end of the intestine when most of the lipid digestion occurs very early? So this is really exciting for us. And the interesting thing is that these receptors, they actually respond to all the different fatty acids present in this oil. But to our surprise, this shorter stearidonic acid, 18 carbon, has shown to be the most potent agonist of this receptor. So it kind of explains some of our findings, and several research groups, they have now done clinical studies with our lipids. And I have just compiled everything into this one figure. They are all from published studies, so we can probably find them in your database, I assume. But if anyone wants the references, I can send them to them. So a couple of the studies actually included moderate exercise in addition to the dietary intervention. And all the studies were done between 3 and 4 months. And we see some clear effects on the improvement of body composition. We see an increase in lean body mass, and we see a decrease in fat mass. Throughout all the studies, we see that the oil has an impact on the insulin sensitivity. I think it's really interesting. We actually see an effect on muscle strength also. And we see an improvement on the pumping capacity of the heart. And as I said before, less than 25% of the oil is omega-3 fatty acids. But we have this significant impact on the omega-3 index also. So all these findings, they correspond very well with what we've seen throughout the years in the preclinical studies also. So this oil clearly impacts the metabolism. So we've developed an entirely new value chain. We've developed the harvesting and the processing technology. We are still developing the scientific documentation, and we're bringing the product out to the market. We have this tiny, tiny crustacean, small in size, large in numbers. It renews rapidly, so it's a sustainable resource. We don't have to refine the oil. The oil itself is liquid wax. And we clearly see some biomedical, interesting biomedical effects. And we have just barely scratched the surface. So stay tuned. Thank you for the attention.
Thank you, Elise. Okay, for our next session, Katrina Benedicto has a 20 years experience as an international marketer and expertise in creating consumer brands emphasizing lifestyles of health and sustainability. In her role as Sustainability and Communications Director for New Seed Nutritional, Benedicto leads in the brand development and marketing strategies for Aquaterra and Nutriterra. She is also responsible for setting the foundation for the team's global sustainability program. Benedicto was drawn to the New Seed's global leadership in progressive agriculture, applying biotechnology to solve environmental challenges. Katrina, let's take it away.
Good afternoon. I'm here from New Seed Nutritional. Perhaps you don't know who New Seed is because we are not typically in the nutrition space. Historically, we are a seeds company, and we are using agriculture as ways to solve huge environmental challenges. One of them being the gap between how much omega-3 is needed and how little is available. So today I'll introduce you to what Nutriterra is. It is our transgenic canola that produces long-chain omega-3 fatty acids. We'll talk a little bit about the omega-3 market and what the opportunities are for the plant-based segment here. I'll give you some highlights from our clinical trial results, and also we conducted research with Qualtrics, and I'll give you a brief overview of our VMS consumer research. Actually, I'll start with this video.
Consumers understand omega-3s are fundamental to overall wellness. Still, 83% are deficient in these essential fatty acids because current supplements don't meet their needs. Many dislike the odor or burp back associated with fish oil. Some worry about marine contaminants like heavy metals and microplastics. Others have environmental concerns, and rightly so. The ocean is under increasing pressure and cannot supply enough omega-3s to meet demand without depleting wild fish stocks. For all these reasons, 64% of consumers prefer a plant-based omega-3 when provided an option. Introducing Nutriterra, the world's first sustainable plant-based source of total omega-3 nutrition, including DHA, EPA, and ALA. We applied advanced biotechnology to deliver the benefits of microalgae through renewable New Seed omega-3 canola. Nutriterra can double the world's supply of DHA on less than 5% of available existing canola-producing land. Unlike algal oils, Nutriterra's source crop is raised on sunshine and rain, making it far less resource-intensive. Canola improves soil and provides habitats for pollinators. What's more, Nutriterra is US-grown, Friend of the Sea certified, and adheres to the highest traceability standards. We document the production process from soil to finished oil to verify quality and purity. Human clinical trials demonstrate that Nutriterra is safe and effective at increasing circulating omega-3 levels, an indicator of health. Plus, it boasts a mild taste and aroma proven to be well tolerated. Nutriterra Total Omega-3 provides an ocean of nutrition within a canola seed to improve human health now and preserve marine resources for future generations.
So why did we do it? Well, we've talked a lot about innovation and sustainability over the last couple of days, and we do feel like this technology is a great combination of these two things. We know that the supply is not really increasing. While sustainable as seafood is a renewable resource, it's not renewable at the rate that the demand for marine resources are increasing. We're looking at a deficit of almost 1 million tons of marine oils by the end of the decade. Now, this technology was developed in partnership with Newseed and CSIRO and GRDC in Australia. And really, it was about seeing an opportunity to use plants to close this nutritional gap. So how did we do it? We took genes from 7 different microalgae and inserted it into canola. So it is a simple plant-to-plant technology here. And I like to think of it as a hardware and software combination. The canola here is the hardware, and within the canola inherent, there was a very high amount of oleic acid. But we take the information from the algae, from the microalgae, and it really teaches the canola how to transform that oleic acid into long-chain fatty acids through enzymatic reactions. And not only are we able to produce DHA, EPA, a total of 7 long-chain fatty acids in here, we also reduce the amount of omega-6 in here, improving the overall nutritional profile of canola. So who needs a new omega-3? You'll notice that the plant-based omega-3 is a small segment of the omega-3 market, but we also know that omega-3s are growing rapidly. From the presentation we saw yesterday on Amazon, the plant-based omega-3s are doubling year over year. So while I don't project that plant-based is going to ever be the dominant part of this, there are consumers that are interested, and it's a way to bring in more consumers into the omega-3 consumption, close those nutritional gaps. There's historic precedence for this. We've got some historical information on dairy alternatives on the left, and plant-based meat projections historically and moving forward on the right. And I would say that at this point, plant-based is no longer niche. It's no longer the hippies in their Birkenstocks eating the granola. It's everybody. Everybody is now consuming plant-based. Not that they're vegans, but there's lots of reasons to do it, whether it's environmental or sustainable motivations, health motivations, or simply taste preferences. Right now, with the younger consumers, 1 out of every 3 persons under the age of 25 will always choose a plant-based milk over traditional cattle. So what do fish and cows have in common? They're outdated technologies. They're still going to be the primary way of doing things, but there's room for more people if we bring in more alternatives, as long as no one feels like they're compromising on their health or taste preferences. The plant-based omega-3 category is projected to be worth about $1.3 billion at the end of this decade, and that's here just in the US. So Nutriterra is applying the power of plants to improve human health and ocean habitats. This product has been clinically studied, and here are some overviews of our human clinical trial. We produced these results in partnership with our friends here at Nutrisource, and while the white paper is currently available on request, it's also being published in Frontiers in Nutrition within the month. It's a two-phase clinical trial to determine the pharmacokinetics as well as the safety and efficacy and the tolerability of Nutriterra. We evaluated healthy adults and gave them doses of 300, 600, or 1,200 total omega-3s and evaluated them over 16 weeks. You'll see that there's dose-dependent increases in plasma of the long-chain PUFAs. So the more you take, the more you get. And over the period of 16 weeks, all of the categories, or all of the groups, dose groups, from low, medium, and high dose increased their DHA, except for the placebo group, which is to be expected. Overall indicators of omega-3s increased. And interestingly, the omega-6 to omega-3 ratio also improved over this period of time, except for in the placebo group. So in context, we didn't see a lot of lipid biomarkers showing significant changes, but this is to be expected because we took a healthy population and gave them a recommended kind of maintenance dose just to see if the product works. And it does work as expected as other sources of long-chain omega-3s. But for people that were on this, we did see that we were able to increase the omega-3 index to move people from high risk to low risk of cognitive deterioration with age, and we were able to improve the relative risk of cardiac events and other cardiovascular health indicators. Importantly for omega-3, we know that a major issue has been compliance, so tolerability was essential to us. Being that we're plant-based, we do have fewer of the marine oil kind of scents and aromas going on. Our participants found the Nutriterra pills easy to swallow, without aftertaste, and without burp back. So having that positive sensory profile made it a lot easier to have people participate, stay in, and have compliance. Regulatory status, because we are a new product with new genetics, we do work with the FDA and other governments around the world. We have regulatory approval in Australia, Canada, and the US, and we have just been granted the status of a new dietary ingredient by the FDA. Last year, we conducted a consumer research project with Qualtrics, and the reason to do this is we wanted to see how consumers would feel about a new source of omega-3s. There were over 80 questions in this survey and over 1,200 current vitamin, mineral, and supplement consumers. Of them, 84% of them are either currently or have previously been omega-3 users. So we're really looking at that 54% people that have taken omega-3s and stopped. We want to know why and what can we do to bring them back in. We also want to know what can we do to bring more people in to the omega-3 consumption. We also wanted to take a look at the younger generations. So about 2/3 of our participants here were also under the age of 40. Here's a summary of the findings that really, what surprised us most is that 2/3 of vitamin, mineral, and supplement consumers consider themselves to be ocean enthusiasts, and no harm to the ocean was actually one of the things that they were most interested in this product. We found that when we asked these consumers, what kind of omega-3 do you want to take? 40% of them said, given an option, I would prefer a plant-based omega-3. Then we showed them the description of Nutriterra Total Omega-3, and that number jumped to 64% of these consumers would prefer a plant-based omega-3 when provided an option. The likelihood to try this product is very high. Over 90% of the respondents recognized at least one advantage of it, and 2/3 of them, no, 3/4 of them, were willing to try this product. One thing we wanted to know is, how do consumers feel about omega-3s? Great news. 81% of these consumers think omega-3s are awesome. They know that they're good for heart health, and with younger consumers, we found that there's actually a keen awareness of the role of omega-3 to support immunity. Bad news, 82% of Americans are still deficient in omega-3. So again, we can go into deeper detail on this if you want to at another time, but what we're finding is that the current omega-3s are simply not meeting the consumer preferences, and having more options will bring in more consumers. We wanted to talk to these consumers about their impressions of environmental concerns related to omega-3, and we had a forced ranking of these 6 concerns. Of them, ocean health turned out to be the #1 concern, and being that we are produced through biotechnology, we wanted to get their sentiments on this technology. Showing the opportunity to rank these, biotechnology was their smallest concern. And what was their response to this product once they were introduced to it? We described it as an ocean of nutrition within a canola seed. Until now, DHA and EPA have been sourced from wild fish and some algae. The ocean cannot provide enough of these nutrients to meet human nutritional needs without depleting wild fish populations, and this clinically studied oil helps fill that gap. This oil provides plant-based total omega-3 nutrition, including DHA and EPA and ALA. Modern plant biotechnology delivers renewable, mild-tasting oil by introducing omega-3 genes from marine algae into canola. This canola is grown on US farms and is Friend of the Sea certified. So how did that resonate? Again, we went from 40% to 64% of consumers now would prefer a plant-based omega-3. And asking them to rank what's the most important for this, they really are very interested in the Friend of the Sea certification, the overall nutritional profile of this product, and the least of the two concerns are actually the mild taste, which speaks really well to what's happened within the fish oil industry to be able to make these products taste better, and biotechnology. So that turned out to not be the concern that we thought it would be for these consumers. So really, what we're offering with NutriTerra Total Omega-3 is a sustainable and scalable technology that works within existing infrastructure. So like we said in the video, we can double the world's supply of long-chain fatty acids on less than 5% of current canola-producing land. No new land is needed, no new infrastructure is needed, as canola has been around for decades, and the ability to process and move this around the world quickly and scale it quickly is a phenomenal opportunity to meet more nutritional needs where people are at. Thank you.
Thank you, Katrina. Our last speaker is Nels Holm, PhD, MSc, is a licensed pharmacist and master and doctorate degrees in pharmacology from the University of Oslo. He currently serves as chief scientist at Aker BioMarine in Oslo, where he has directed and participated in preclinical and clinical research, as well as in process and product development. Holm has also been involved in basic research into Antarctic krill biology and ecology in Western Antarctica, where krill is harvested. We welcome Nels Holm.
So thank you, and thank you for this opportunity to talk about our lysophosphatidyl-choline products and the research and product platform Lysovita. I will have to go into somewhat teacher mode and go back to the basics to show why it makes sense to focus on lysophosphatidyl-choline rather than phosphatidylcholine itself. So you have to bear over with exactly that. So I will take you through the basics. Then I will present you for a paradox that we might not think of when it comes to lipids. Then I have a solution. And then, of course, I present you with a conclusion. So let's go really back to the basics. Let's look at the definition of lipids. This is something that most of you would take for granted and never really think carefully about. But it's very broad. So let's have a look at what the Oxford Learner's Dictionary says. Any of a group of natural substances that do not dissolve in water, including plants, oils, and steroids. That's not a very specific chemical definition, I would say. Then we go to the Collins Dictionary, and I actually get it wrong, but that's a different. But any of a large group of organic compounds that are esters of fatty acids, well, that's wrong. We should tell them. Or closely related substances, usually insoluble in water, but soluble in alcohol and other organic solvents. So there we go again. They are important structural materials in living organisms. So let's go to the British. From Encyclopaedia Britannica, they say that any of a diverse group of organic compounds, including fats, oils, and certain components of membranes, that are grouped together because they do not interact appreciably with water. Very British. So the only common denominator of lipids as a chemical class is that they do not dissolve in water. And that has profound consequences for how an aqueous body like our own handle these. So let's have a look. So First, I would like to show you something about lipids and water solubilities. I made a list of all carboxylic acids from formic acid and all the way into the carboxylic acids that are fatty acids that we happen to call fatty acids. And here we are. If you look at the X log P column, that tells you something about how much those molecules likes lipids, that is octanol, or water. And only the shortest ones likes water, they're water soluble. And pretty soon when you elongate those acids, they will prefer octanol. And as you see, some of them are absolutely insoluble in water. There are 1 trillion times different, more of them in the octanol than in water. And the fatty acids, usually we start to define them by an X log P of 3.0, so by caprylic acid. Now, if you look at the fatty acids that is the object of this meeting, the EPA and DHA and DPA, then they are hover around 6 to 7. So they are insoluble in water. That presents a few problems. Now, first of all, this has already been done in this, but still we have a look at what this means as a diverse group of molecules. And I'm going to focus in the continuation of this on the glycerolipids, that is the neutral glycerolipids, TGs, but first and foremost on the phosphoglycerides. Well, one thing, when going through this, I realized that the Oxford English Dictionary Word of the Year 2021 was VAX. Well, almost, that is. So, what about, we need to go back to some basics on glycerolipid digestion and fatty acid uptake. And this is kind of textbook stuff, but textbooks aren't always forthcoming with reality and what we exactly know by now. So, all of you know that digestion starts with intake of, in this case, lipids. It's blended, made into smaller spheres. Then the enzyme comes in, both from the mouth, from the gut, from the stomach, and from the pancreas. And they are a bunch of separated molecules by now. They still don't like water very much, so they need to be emulsified. This is the first step of their uptake. And after emulsification, they are further acted upon, because by the emulsification you get a much larger surface area that the enzymes can act on. So then the end point of that is making this into a mixed micelle, which are tiny little tight micelles brought about by bile salts. And from then on we can liberate free fatty acids, monoacylglycerols, and cholesterol. And then by diffusion or by fusion into the membrane, it can be taken up into the enterocyte through the brush border, resynthesized, combined with ApoB, made into kilomicrons, then dropped into lymph, and from there on entering into blood and then all the tissues. So first lymph, then tissues. This is the standard story, and it's a very complex process just for the digestion and the acute uptake. Now, what about short-chain FFAs? Well, they go straight through. They don't need resynthesis. They can be taken up directly, and then they go into blood, exactly like the bile salts, and then brought to the liver. So, what about lysophosphatidyl-choline? The cold phosphatidylcholine that has been left with one fatty acid only. Well, papers from the 1980s, 1990s actually demonstrate that they can also be absorbed through this direct route into blood. So instead of entering the body through lymph, they can enter our bodies through the portal circulation. So they go directly to the liver. So there are two different uptake mechanisms, and this has vast consequences for their further processing in our body. Now the paradox then is that mass transport of lipids in an aqueous organism actually has to go through water, lipid barriers, water, lipid barriers, and this process is dependent on the metabolic stability and directed metabolic transformation. It's dependent on water and lipid solubility, protein binding, concentration gradients, transporters, and micellar structures, that is lipoproteins, for example. So if we have a look at what this looks like, it may be fast. In some pools, it might be very slow in other pools. The important thing, let's go back. The important thing is that the overall process will never be any faster than the slowest process in the chain. And here, this is where solubility comes in. So what about if we put in transporters? So we can put in a transporter in the membrane, or we can put in a carrier in the aqueous phase, and they will help entering that particular stage, but if there is an insoluble step in between, the overall process will not be any faster. This has consequences for the total mass that you could drive through the system. So let's have a look at real data on the uptake of DHA, and in this case administered orally as phosphatidylcholine. What we did was to synthesize this molecule, and we put in a C14 in the carboxylic group. So it's a radio-labeled experiment, and of course then we have to do it in the rat, and you have to forgive me that I've put a human in, but believe me, it's pretty much the same. Here we have the first 24 hours. So we take the substance, it's being digested, and then we will have the uptake curve. So this is in blood and in plasma, and the dots you see is the observed concentrations, and the lines are a mathematical model describing how this goes on. Now, look at this. This is the first 22 hours. So keep that in mind. Then let's squeeze it together. So this, and then we followed it for another 2 weeks. And it looks like this. So it takes one dose, you have one little pill of, or you have one meal of salmon, and your body is handling this transport-wise for 2 weeks. And recognize the undulations. And these undulations are exactly the same in the human. It's a very complex in and out of the systemic circulation situation. Now, this is a graph that I found in a paper from Kagan and co-workers from 2012 or 2013. And these are fats from C14 up to actually alpha-linolenic acid. And they're taken up in an hour, and they're gone in 10 hours. So let's put that in perspective with EPA, or DHA in this case, but EPA would be pretty much the same in this form. It's two different worlds. And what this should tell you is that the handling of these molecules are very, very different. So the fats we eat for energy are handled completely different from EPA and DHA. So what about the brain? Well, as you know, it's a fatty organ. It consists mainly of sphingolipids, glycerophospholipids, and cholesterol. It sits in the membranes. So it's made for structure and signaling. It's rich in both, very rich in LC-PUFAs, and it has a relatively high turnover rate. So how does lipids enter the brain then, given this insolubility problem? Well, first of all, in the brain, membranes are localized. Membranes localized fatty acid transporters are very important for shuffling them around the brain. And there's a lot we don't know about how lipids flow through the brain. We have the lymphatic system. We even know that there is a lymphatic system there. But there's quite a lot that we still do not know. But we know quite a few of the mechanisms that is probably involved. Now, how does PUPAs cross the blood-brain barrier? Well, the first clue came, well, there were previous clues to it, but Michael Lagarde and his group published in 2000 a paper that suggested that what is taken up into the brain is not the fatty acid, but lysophosphatidyl-choline with the fatty acid on. And that was confirmed in 2014 by David Silver and his group when they published the letter in Nature on MFSD2A. Now, there is an extensive research on MFSD2A since then. Its functions is to transport fatty acid, but it also is important for the integrity and actually the formation of the blood-brain barrier itself, and also the blood-retina barrier. And this is just where it's expressed. It's expressed in the brain, it's expressed in the liver, it's expressed in our genital organs, and it is expressed in the placenta, and then a few other organs. Skin is kind of an outlier there. And this is what it looks like. Beautiful, beautiful molecule, very well understood by now. Let me go back. It's now called the sodium-dependent lysophosphatidyl-choline transport symporter 1. If you could say that one, you can have another beer. And this is how it works. It's a classical flip-flop. It's co-transporting, it's driven by sodium gradients. And it has a 12 transmembrane alpha helix. It's the largest hydrophobic pocket known in nature. So at the very end, I would like to show you this one. This is lysophosphatidyl-choline DHA that we've injected. So if you thought that these undulations that you saw over the first hours after ingesting PC DHA had something to do with absorption, you're probably wrong, because you have exactly the same kind of undulations when you inject it. And look at this. It flows out of plasma and blood in the first half hour to the first hour after you inject it. Then it comes back in a few hours, then it goes down, it comes back again, and actually it starts to come back at 22 hours again. And you can see the whole profile in logarithmic form on the top. Now, so we propose that there is a sixth form of LCO3 molecule. Together with the other ones, we should view LPC as one of the key forms that our fatty acids takes on. And finally, it's this transport stupid, and it's a light play on what James Carville, the strategist in Bill Clinton's campaign in 1992 said, when he said it's the economy stupid. Now, finally, Lysovita is a commercial platform, but it's also a research platform, and we are more than interested to work with others to elucidate and work out the ins and outs of exactly how lysophosphatidyl-choline works when it comes to both transport, but also accumulation and uptake of lipids. Thank you.
Thank you, Nils. Can we have our speakers back up on the stage, please? We have time for a few questions. I think we have some runners out there with microphones, so. Anybody have a question?
I have two small questions. So my first question is for Alice. For the species whose name I'm not going to try to pronounce, you said it's at the bottom of the food web. Have you kind of assessed what the impact is on the whole food chain if you were to start harvesting maybe large quantities? I'm just curious from a sustainability perspective what that means.
If you remember the giant circle of just the annual biomass, and the Norwegian government has issued this management plan, which is 0.05% of that total. And we know from biology that only 10% of the energy is transferred from one trophic level to the next, which means it's actually 90% just falling to the bottom of the ocean. So I assume we're on the safe side there.
Okay, got it. Thank you. And my second very short question is to Katrina. I think when we talk about canola, you said you won't need any new land. Does that mean that this high omega-3 canola is displacing food-grade canola? I guess how does that work in terms of land use in the U.S.? It sounds like you're located.
That's a fair question. So in some places it might be displacing a canola. In other places it might be used as a rotational crop. Canola has a very deep root stock, which actually helps to aerate the oil and provide habitats both in the soil and in other areas. So it's beneficial to the farmer to increase this. What we found in areas where we're planting canola where it had been fallow in previous seasons is that the grains that are harvested after a canola crop actually increase the yield of the barley, the oats, or the other cereal grains that are planted after a canola. So we do see this as a net positive. It may displace some canola, but overall it's still a very beneficial product.
Thank you.
Any other questions? Oh, there's one.
Hi, question for Katrina. Since canola is an open pollinated crop, what is the thought or planning around, I guess, segregation of a GMO canola from the rest of the supply chain that would be non-GMO?
Well, our product is certified through ETS, that's Excellence Through Stewardship, which is the biotechnology supply chain management. Everything is traced very, very carefully. In the places where we're planting, we do maintain really good crop barriers. In addition, we ask that farmers not grow another type of canola on the same subsequent land for at least 3 years to prevent any volunteers, as we like to call them, of these crops coming back up. So we're not finding a lot of issues with low-level presence.
Question over here.
Okay.
This question's for Omega. Interested to know if you would share what your next ambition is for a clinical trial using your material for possibly some other application.
Well, we, like I said in my talk, we're focusing now on extreme premature children born before week 28. We also have another pipeline that I can't share, fortunately, since we're now listed. But there's lots of good stuff to come. Yeah.
So Hogne, I have a quick question as well on your product that you described to be based on SPMs. Can you comment on the concentration of the SPMs in your material?
I can comment that we think they are clinically relevant. There's a lot more work to be done on SPMs, both in terms of composition, quantification, and also the broadness of what we find, both in terms of PRMs and also the individual SPMs. So I can't say on the picogram level what they are, but we do have standards for some of the SPMs that we know the exact amount, but there's more work to be done to verify both, batch to batch, but also in terms of the composition between both PRMs to SPMs and also the SPM to SPM, if you know what I mean.
Yeah, thanks.
Go ahead.
Yeah, I can't let Niels get off without a question. So I've got two questions for you. One is how lysophosphatidyl-choline is transported in the bloodstream.
We don't know exactly.
Okay.
It's a tiny fraction at any time. And it doesn't mean that all that is taken up as lysophosphatidyl-choline stays that way. It's definitely, and probably because, the transformation, the undulations you see in both plasma and blood is probably a reflection of the very complex transformations that takes place.
Yeah, that was my second question, because I think you labeled the DHA, right?
Yep.
So I wondered, when you had those undulations, whether it was always in the lyso-PC form, or whether lyso-PC was going somewhere and DHA was coming back out in a different
I wish I could
complex
answer.
form.
I wish I could answer that question precisely.
Okay.
What I can say is that you see undulations both with TGs, you see undulations with LPC, and you see undulations with PC also. So it's a general feature. Also, it's not specific for the rat. You definitely see it in humans the same way. So it's just a reflection of the very complex caretaking, I would like to call it, of EPA and DHA as fatty acids. And lysophosphatidylicholine is just one way of taking care of it. But my point is also that it is way more water soluble than the fatty acid. It's 100 to 200 to 300 times more water soluble than the fatty acid. And it kind of solves then the paradox that we shouldn't be able to move these around at all.
Anyone? Charles? Yeah.
I had a question for Katrina. Two questions. One's multi-part. If you planted an acre of this canola, how much canola oil would come from that acre? And then of that oil, how much is EPA and DHA? So that's sort of one question. The second question, very practical question, what do you do with the spent organic material?
Excellent questions. So I'm not going to get to our exact yield numbers because it is a newer technology. We're working on increasing the yield. It's comparable to conventional canola in terms of yield. Our canola oil is 33% total omega-3 when we extract the oil. So what that means is with the DHA and EPA, we're looking at about 11% DHA, EPA. We've doubled the amount of ALA available within the canola oil inherently. And for a DHA equivalent, we say that 1 hectare, which is 2.5 acres, replaces the equivalent of 10,000 1-kilogram fish. So with our first year in the aquaculture business, we feel that we have saved at least 600 million fish to be able to not be fed to other fish. Your second question was, what do we do with the leftover material? What we have left over is meal, and that goes into feedstock for cattle and to chickens and other livestock type animals. So there really is no waste there. And then any of the roughage that stays on the land actually gets put back into the soil to replenish the soil and build that back up.
I think we have time for one more question if there is any. Otherwise, going. All right, well, please join me in thanking our speakers today.